Good afternoon, everyone. Thank you for taking time to be part of the special seminar. Today we have our guests, and I'll do a brief introduction of our guests shortly. But it's nice to see a full house. So, Professor Peter and your team, looks like a lot of anticipation towards your talk. So, the expectations are very high. All right. So I take immense pleasure in introducing the team that has come from Russia. So they are from Sechenov University. Professor Peter Timashev, please. Yeah. Dr. Alexei Fazulin. Then we have Dr. Pauline Molina. Big Molina, that's great. And Dr. Yana, yeah. So, all four of them, we wanted all four to talk, but it's not possible to squeeze it within the 1 hour time period. So they have decided one of their team members will talk. But prior to that, I just wanted each of them to give a brief outline of the work that they're doing and where they come from. As means of introduction, Professor Peter Timashev is the scientific director of Sechenov Biomedical Science and Technology Park and a professor of a world-class research center on digital biodesign and personalized healthcare in Sechenov University, Moscow, Russia. Dr. Alexei Fazulin is the head of Laboratory of Digital Microscopy Analysis, Institute of Regenerative Medicine in Sechenov University. Dr. Paulina Bigmolina is the head of Design Center of Biofactory Institute for Regenerative Medicine in Sechenov University. Dr. Yana Kritis is the head of Laboratory of Regenerative Veterinary Medicine at the Institute of Regenerative Medicine, Sechenov University, Russia. So the colleagues have been from Russia have been here for a week, and they have already ran a multi-institute workshop on photobiomodulation. The workshop took place on Monday and Tuesday. We completed the workshop, and now they're here for the 3D bioprinting seminar. And I know it's a topic that is of quite a bit of interest. So I'll let them speak, and I'll start. Professor Peter.
Okay. And of course, it's a great pleasure to be here. It's my second visit to South Africa. The previous was about 20 years ago, when I was so young, and I lived in Stellenbosch for 1 or 2 months, I believe, and really enjoy the beautiful country, the wonderful people, and fancy ocean. Just now, yes, we're working in Sechenov University. Sechenov University is a leader in Russian medical education. We have 20,000 students focused on all issues about medicine, medical engineers, surgeons. So everything dealing with medicine go through our university. And probably about 10 years ago, Institute for Regenerative Medicine was started, and I've been invited by the people from university to give a scientific expertise in the field of biomaterials. And from that period of time, we grow large, Just now, Institute for Regenerative Medicine, it's about 120 scientific fellows. And we have 3 main topics we are working with. The first is biomaterial. Biomaterial, we try to make it I like this word, robust, very cheap and easy handling, and we have a very good success in that. We created some product that you can buy in Russia in every pharmacy. It's hemostatic sponge. Just now we're working on material to restore skeletal tissue made from collagen. So this is one of the very close to clinic direction of our research. The second focus is biophotonics, includes photobiomodulation. We work, Polina's work is very close to this field. Photobiomodulation is not so popular in Russia like here, because I understand that there are several groups working on photobiomodulation in Russia. It's not so popular, but we also created our first product. It's setting that allow us to deliver light with certain wavelength and increase the wound healing, enhance the wound healing, and we have different utilization of this device. And the third direction is bioprinting. 3D bioprinting. This year will be first clinical translation and utilization of bioequivalence in clinical practice in Russia. The focus is skin, also cartilage, also vocal folds and ear drum. Just restoration and scar removing. So these three main direction of our institute. Recently, we have a very good lists of publication. Every year we publish more than 50 or 60 publication all over the world in different scientific issues. So I believe we understand in Russia how to move from idea to direct to clinical practice and bring some good stuff for our patients in Sechenov University. So we decided that, as you can see, our team is really young, 25, 26, 27, sorry, 27 years old. Yes, 27, 30, 25, 27 also. So so all institutes and the guys already be head of the laboratory. It's because they're really talented, and it's very interesting that we travel with this multidisciplinary team, because I'm a chemist, Polina is biologist, Alexei is clinician, yeah, and Yana is veterinarian. So together we bring, wonderful expertise for this field of research, and Polina will present now her brilliant results. Thank you.
Thank you for this introduction. You hear me well now? Okay. And can I have some laser pointer, or if not, it's not necessary. Okay. Okay, great. Yeah, dear colleagues, thank you very much for welcoming us here in Cape Town. It's really was great pleasure, and it is a great pleasure for all of our team. And we're sorry for the delay. Unfortunately, we had some hard time climbing down from the lion's head, so it was really nice. And today I will try to return my mind back to bioprinting, which is also very interesting, but maybe in some other field of feelings. And as Professor Peter said, yeah, we have a lot of disciplines in our institutions. And for now, our main idea is to connect all of that to make some standard track for the research to been researched in the lab, then tested, then produced, and translated into the clinics in the most like fast and maybe convenient way. And as my main topic is bioprinting, today I will focus on it. And also I will try to highlight some interesting hypotheses, findings, and things that we have faced during our years of research in that field. Sorry. So we can start from the basics, because I'm sure that everyone know what is bioprinting currently, but usually it is really comfortable and simple to make some associations with the FDM printing, 3D printing. This is the normal thing that everybody knows. 3D bioprinting is quite the same time, but here we use not plastic as a bioink, as a filament, but the bioink, which contains usually hydrogel and living cells. So we are printing 3D living structures. And as for 3D printing, all starts with a 3D model, which then can be sliced and produced back to the machine to produce something in solid shape. Also, it is just an interesting insight from history that the bioprinting began somewhere in at the age of the millennium, and it started from the Thomas Bohr from Clemson University. Also, I'm sure a lot of people know that man. And interesting part is that they started to work together with Dr. Vladimir Mironov. He is from Russia as well, and he has some really interesting papers highlighting the development of this field for the 20 years ahead of his time, as we can see it now. And sometimes it is really needed and convenient and insightful to go slightly back and to read, the founding fathers, to have some initial ideas and see how they now are implemented in the real practice. So just again, to not be confused in the set of our following works, usually for the bioprinting, you need two general things. It is the bioprinter itself. It can be laser printer, extrusion printer, some in situ bioprinter, different type of machine, which produces 3D structures out of your material. The material, the bioink, is the second part. And here we are working primarily with the living components. I'm sure you can know what the structure is. It could be the cells or the spheroids, organoids, some multicellular three-dimensional aggregates of cells, like more complex parts to build a tissue. And of course, you need the hydrogel, because it will be act as a microenvironment for cells to migrate, to communicate with each other, to proliferate. And also it is the mass of your bioink, because it is usually viscose and provides the necessary shape and fidelity for the printing structures to preserve the initial 3D model things in the solid form. And for now, we are getting the third component, which we call biopaper. Usually it is a membrane out of some proteins or maybe polysaccharides. It doesn't matter in the detail. The idea is that you need to have some 2D membrane, which is elastic, and you can print onto that, so that it will be much more easier for clinicians, for example, to handle it, to sew it to some surgical sites. And also, usually, it is much more biomimetic, because usually our organs and tissues are covered with the capsule, and this thing, biopaper, can mimic that capsule, so it allows us to produce more histologically relevant and complex structures. And it is actually the main idea of my presentation today, and also I think the set of our research works currently, because we have some different methods, collaborating groups, some expertise in different fields. And for now, we are trying to build it up all together, starting from the digital things such as medical data, because as I said, the bioprinting starts from the 3D model. And here you can use the for example, CT scans from the patient as a primary model to produce some relevant things after that. For that, you will need, of course, some software to slice it. It is a lot of software available for free currently, but it is not usually adjusted to the bioprint because of some minor technical things, different models, different dimensions, all of that. So it is the things we know how to do, but don't have at the fingerprints yet. And also because this is actually one of the topics of today's talk, but I will touch it just a little bit. It's the machine learning and artificial intelligence, because as we discussed previously at our seminars and workshops, it is also a very useful instrument. You should know how to use it correctly, and sometimes you need to have constant feedback to make some correct decisions. But still, bioprinting is the field where it can be applied quite wisely. I will return to it back a bit after. And of course, we need to have biomaterials, bioactive factors, which can, for example, help cells to differentiate into some specific lineage. When we need to produce a bone, we can add some ceramic particles or some chemicals so that to push them slightly, like, hey, we need a bone, please, not cartilage or not adipose tissue. The cells, organoids, and multicellular things. Also, I will stop here a bit because maybe the wise and accepted definition of organoid usually is something very expensive, very hard to produce based on the iPS stem cells. But the field of bioprinting currently is widening this definition, because it has shown in our lab and in many labs all over the world that the primary thing that should be during the bioprinting is the 3D structure of spheroid, for example, because cells are more densely packed, they are compact, they are more stable, and they can live in the much amount of stress. For example, in the shear stress, they're much more, I forget the word, but they are better than the just ununited single cells. And if you will mix some different types of the cells, for example, if you even get some cell lines and add to the mesenchymal stromal cells the HUVECs, which are vastly recognized, they will build some synergy, and you will observe some interesting results of making up the capillaries and some vessel-like structures. So that is why we are trying to use now these 3D assembloids or organ building blocks with different types of cells, so that they can self-organize, and it provides us usually more interesting and inspiring results after that. After the bioprinting, also the printed construct should be mature, because when you print cell, it's just in the stress somewhere in the hydrogel, doesn't know any signals from the environment and all of that. So they need some time to adapt, to interact with each other, with the hydrogel, all of that. That is why we are now currently working on the producing of the bioreactors, some specific things for specific tissues. For example, for cartilage, it is better to apply some constant pressure so that the cells will be more used to the stress that they are going to be in the native organism. For the skin, for example, you can stretch it a bit, also to provide some mechanical pressures. For the vessels, it would be natural to implement a perfusion system, of course, so that they will function, not only just be in hydrogel, but start to build some functions. And of course, one of the important current problems is the evaluation of the printed and matured things, because they can be really different, even in the same conditions of the same experiment. Cells sometimes self-organize into new structures which you do not expect to see. And of course, if we're talking about the translation to the clinical practice, we need to have some methods to control the outcome, our product. And this is why we're working now densely with the biophotonics, because it is the interesting approach to first of all, to non-invasively check the status of the cells, their viability, metabolic activity, some of these parameters. And also, it is very resolutional method, very informative set of methods, I would say. And so that is the main idea, that we are trying to tie all of the things up to the producing of the some tissues and organs analogs for the transplantations, or as models to test some hypotheses in vitro. And here I will start to show you some real results. For example, at the slide, you can see just the examples of our bioprinters. How do they work? As you can see, it is quite a simple system. Yes, they have the sterile chamber to make it more sterile and to control temperature. But actually, this print head of the bioprinter is itself 3D printed on a 3D printer. So, and also I need to say that it is the different, very quite novel and very perspective direction of the bioprinting. It is called in situ bioprinting. You can see, for example, some of the our models that we are working on. And the idea is, maybe you have seen in your life the 3D printing pen. It is just ejects some material and lights it up, and it becomes dense because it is cross-linked. So the idea here is the same, but again, instead of plastic, we're using bio-ink with the cells. And this is very actually interesting field, because we can use not only the UV light for cross-linking here, but for example, the photobiomodulation wavelengths, which would be beneficial for the surgical sites and the wounds that we're working with. Also, last but not the least, but it is some about really thin matters, but biophotonics also have its place in here. In laser bioprinting, when you use the single light beams to like print, to push some cells or spheroids, some particles with the extremely high resolution. Usually it is needed to test some interesting hypotheses about to how the cells interact, and you can like provide some structure, some pattern in a high resolution, and then observe the results on the like single cells or spheroids. Oops. And here I will also add some good words about the biopaper. For now, we are primarily using collagen biopaper, so it is 100% natural, I may say, and it was really good for the interaction with our doctors, with our clinicians, because hydrogels are really hard to operate. They're, you know, like a jelly, and you need to take it, place it somewhere. But with this biopaper, it's much more natural for them to use more dense membranes. And this project, for example, we have printed the hydrogel onto the surface of the collagen paper and then differentiated it into the cartilage lineage, so the 100 differentiation. And then it was placed into the knee of the rabbit to see how the structures containing biopaper, hydrogel, and cells would benefit their regeneration. And as I know, they are currently at the vivarium, because we are using long periods like 6 months and 12 months in that. So this is quite a novel project, and we're looking forward to the results. And here I would also just provide some insights on the like the general idea of putting cells into the bioprinting model, because when you are testing some hypothesis with the 3D structures, and you need to prove something, and you don't have any good, correct working methods, you need a lot of samples. And here bioprinting is the game changer, because you can just model like, yeah, I will have the well with 24 well plate, or 48 or 96. It just depends on your protocol, which you are changing really like in the laptop. A tricky thing always is the hydrogel, because on the one hand, it should be biocompatible and good for the cells. On the other, it should be printable and preserve its fidelity. So usually the thing which is good at one hand is bad on the other hand. So it is always a delicate balance between the printability and biocompatibility. But here, for example, for the mix of gelatin and fibrin, we have worked out on this composition to make these simple grid-like structures. And as you can see, when we place the mesenchymal stromal cells inside, they're starting with this just spheroids, not really feeling well after the shear stress and UV cross-linking, but after 7 to 14 days, it depends really on the type of the cell, of the source of the tissue. We can see how do they grow these sprouts, and you can see here it's the part of the printed construct, and the cell density is quite big and high, and so it is most likely to be some tissue, not only printing some cell printed in the hydrogel, but something that is more about to be a tissue. And of course, we have shown how do they differentiate. So generally, it was the model from which all of the other projects have been started. Very interesting finding in here was that really cells are different. You know, the problem of the donor to donor variability between the different primary cultures. And here we have shown that when you make this simple distinguishes and you put them into the spheroids, then you print, then you mature them, you can achieve two completely different morphologies, physiologies, metabolic profiles, all of it. But it's from the scientific, from the literature point of view, these are the same cells. So you need to take it in your mind. And about the in situ bioprinting, again, you can see here this portable in situ bioprinter. So we used this work with the MSc as a base to provide to work on some hydrogel containing not only the cells but also the bioactive factors. And here we have used the secretome of the cells. So when your culture cells on petri dishes, they are constantly releasing some exosomes, some growth factors, some signaling molecules. When you collect them and you concentrate them and add to your bioink, you have this the language of the cells in your bioink and in your producing tissue equivalent. So it is usually much more beneficial for this adaptation period of cells in the hydrogel. Also, another project about the skin I should mention. This is more, I think, about not producing the transplantants, but producing the models to test drugs on, for example. And here we tried to go in this philosophy of mixing everything together. And we mixed here the keratinocytes and fibroblasts to provide these spheroid 3D structures. And also we added some bacteria in here because on the healthy skin, we usually have a lot of bacteria there dwell in different niches. They, just as the eukaryotic human cells, they have its community. So we have seen what if we will try to recreate this community in vitro on the microfluidic chip using this simple air-liquid interface system. And we have found out that when you add bacteria like in your conscious to your cells, they usually do not die, but sometimes they even grow better, because for keratinocytes, it is essential to have some signals which should lead them to death, such as air-liquid interface, but they actually start to differentiate into the normal keratinocytes that we have on the skin. And adding bacteria, we also try to even enhance this effect. Another example of the same idea, actually, but on the different object, is the urethra. It is quite a long project. It was started a few years ago on the different type of the equivalent, but for now we're trying to also implement this philosophy of having the biopaper and then the hydrogel consistent here of two different layers. The first one is mesenchymal and the second one is epithelial. Epithelial, I think it is quite obvious because the inner part of urethra should have some epithelial layer, yes. But also one of our philosophical ideas is that if you add some MSCs to the mix, it won't be bad at any point. It is always better because it is the idea of the mesenchymal stromal cells to support the other cells. Yes, they are different. They are sometimes not obvious to think of their differentiation and the final and bioequivalent, but usually they're really helping the other cells to adapt with 3D conditions to form something really much more resembling the tissue, such as here, than just, again, the spheres in the hydrogel. This is, I think, the most complex project in terms of organoids and this OBB for now. Here we tried to produce the liver model, which would consist of 3 types of the cells. One type is for the liver hepatic specific cells, HUVECs, because the liver is highly vascularized organ and vascularization is essential for any bioequivalent. And again, MSCs, because why not? They help the HUVECs and they're usually better for everything. And also another important point that we had here is that we added the powder produced from the liver ECM, so the extracellular matrix of the liver, freezed, minced, and then make it to the powder and add it to the standard composition of the hydrogel. And actually, we have seen that, first of all, if we compare just organoids and organoids in hydrogel, it is better in terms, like they have the space around to spread their sprouts to make these vessel-like things. And when we compared it in terms of the specificity of the liver, it is a long list of parameters that should be controlled because liver is extremely complex organ. Here we tried to, first of all, see how the cytochromes of the liver are produced, and we have seen that they are preserved. Not really high signal, but still they are in there. And when we add some DSM powder, it allows us to increase this expression amount a little bit. And another important thing is the drug sensitivity tests, when we have added some specific drugs to our model and have seen the sensitivity. And here we can see it's bad color decision was apparently, but this is the line for the just organoids, and this is the bioprinted organoids. It is quite an obvious result because here they have these sprouts which would provide the inner part of the spheroid with this up is acetaminophen, like the paracetamol, I think. So it should be toxic to the liver. And here we can see that the same cellular component, different matrix component, and the sensitivity is higher in the bioprinted model. And last but not least, I think also our quite a new project, and we decided to go even more in terms of mixing cells. Our object here was the periodontal complex, because it is really hard thing to do to take the teeth inside their place in the gums, especially if you have some infections in your mouth. It is really a sensitive organ and really fragile, I would say. And on the other part, it has a lot of structures in here: the bone, the tissues of the teeth, the dentine, the enamel, and also the epithelial layers and the capillaries, all of that. So for now, we are working on isolating the primary cultures of the cells out of the different parts of the periodontal complex and the gum, then characterizing them because they're very different among themselves and also among the donors. And then again, producing the different variations of these organoids, as we call them, to see which composition would be the most resembling some parameters and properties that we see in vivo. Because this was all about some scientific, quite novel projects, but also for now we have some examples of bioequivalents which are in this time are translating into the clinic. As Professor Peter said, it is the vocal folds for the people with the dysphonia, with the problems with the voices and with speaking. It is the eardrum bioequivalent for the people who have perforations and who developed so they cannot hear. And of course, skin, because it is really like a wide amount of people in every society who needs to to have some skin replacements. And the idea is generally the same. It is different mixes of the spheroids, some adding some MSCs, some membrane, and bioprinting. And then we have the bioequivalents. And for now, it is only the question of time because the bureaucracy is quite high in Russia as well. So we're working with that currently, but it is, I hope that in coming days, finished products and started to apply in clinics. And speaking of this diversity, it is really important to think some steps ahead because there are a lot of different types of hydrogels. The cells which are vary their properties between the donor, the type, the site of isolation, the age, all of that. And also when you combine that together and it starts to mature, it is really long, hard, expensive, and not usually so beneficial to test it all on the in vitro in the wet lab. So for now we have started some projects, the idea of which is to make some general laws and rules and some presumptions based on the acquired experimental data. And as a base for that, we used this database for BioInk. So the idea was quite simple, just to read the papers and put some important parameters, the key ones, and just to have some heat maps. It is not even the software, just the interactive site when you can compare fast the two different types of the hydrogel and see usually which one is better for you, just clicking on the site. So we are a bit developing this idea further to prolong it, not only from the choosing of the BioInk, but also how would they interact with the cells and how would they mature and provide us with some viable bioequivalent or not. But it's still also in just started, and we're still collecting some interesting ideas because there are a lot of them just in the air usually. And also to add something just about some interesting practices in bioprinting. In this field, we don't have any particular expertise, but maybe we're looking in these directions. One of that is 4D bioprinting. Basically, it's the 3D printed structure which is able to change its shape usually or some different properties in response to some signal from the outer environment. Usually it can be the change of the temperature, the change of the ionic power, like the ions in the solution, some light, of course, maybe some sound, all of that. So the idea is this is really beneficial for the organs with a complex structure, which is really hard to place somewhere in the native organism, such as a trachea, because as you know, it has the round shape, but it is like as you see, and to put the whole construct inside the tracheal site would be really hard, I would say, with the high invasiveness. So maybe using some polymers that are simpler in the shape and then they will fold as you want them to in response to temperature would be really better and beneficial. Another really interesting thing in connection with the biophotonics actually is the holographic bioprinting. The idea is that as the STL printing, I think, or DMP, but here you have a lot of special mirrors and of course the high intensity of the laser, and so you shine the UV laser on the different planar planes simultaneously. So you can just economic on your time, not do the slice by slice, layer by layer, but just all in one. And as you can see, they have printed all of these 12 structures, one around 2 centimeters in 2 minutes 70 seconds. So this is quite a good result. And only problem for now here is that it is really hard to find some really biocompatible resin which would not harm the cells during the process. And also here to say about some modeling and this AI stuff is curated bioprinting. Also, the interesting concept based on the some mathematical modeling before the bioprinting. The idea is that you model your laminar flows, not just flows of the bio-ink, with the special mixer. And what is the benefit? At the output of your nozzle, you have preformed layers, like 15, 20 of them. So you have really thin and tiny microstructure, and you do not need any some fancy, really high resolution as is laser printing, for example. You can use some normal extrusion by a printer with a wide nozzle to not harm the cells, and just use deep preformed by ink in layers. And also, this is not really a scalable technology, but really interesting in terms of biofabrication, the Kenzan method. The name comes from the Japanese technique, Kenzan, when they put the flowers and some flowers in this special place to hold them. And as you can do with the spheroids, when you have some tiny needles and you put your spheroids on the needles, you know, we have this dish in Russia, maybe you have as well, the shashlik, like the kebab, but barbecue, yeah, barbecue actually from the spheroids, but you would not fire it. You will just put it in the incubator, and after a while they will fuse with each other, because as I said, it is highly vital and self-organizing structures. And you can see that it is something like a vessel with quite a big scale. So yes, and of course, rolling back to in situ bioprinting, it is the future that everybody is so in this the fifth element film, like something fancy machine printing the human. But for now, it is more like this, as I said again, in situ bioprinting. But I believe that it is the first step, because there are really a lot of challenges to bioprint in humans from the all points of view: technical, ethical, medical, biological, all of them. But these things are developing quite fast, and they have shown their potential, especially for the skin wounds in the diabetic foot ulcers, because it's really harmful for the people, really hard to regenerate. And some combinational therapies usually work quite good in some patients, not all of them, unfortunately, but and this gives us the idea that in the future, we will maybe have some stem cell factories or biofactories where at the beginning you put some hydrogel, some solutions, some cells from the patient, and then all of the machine based on the artificial intelligence will just put it together and just take you some artificial organ for you. It is still in the future, but it is always good to have some really big and nice thing ahead to go for it. And there are some, how would I say, proof of concept currently that this strategy maybe not really have the place in the real life. Of course, as the most ideas. But there are some companies that are integrating all of these technologies together, and it has shown that, yes, it is possible, yes, with a lot of step-asides and corrections, all of that, but it is the normal process of development, something. So I would just return you back to this slide, and that is why we're doing it in the Sechenov University, and we're trying to make some flows of different projects to base on some really global and simple strategies, methods, and ideas. So yeah, and very important point in this slide is that not only this slide does not contain really important resource, which is of course the humans, because it is still done not by AI, by the hands of the students, PhD students, researchers, all of them. So it is really important to spread these practices, to get them to the students, to teach them, and this would also beneficial for us, not only in the terms of experiments, but also to spread some global ideas that we believe on. And this can be done only through the teaching. That is why we are making some, events where we make a model of experiment with the students to teach them these initial philosophies. So I would finish at that point, and all of the things are done, of course, with a great amount of people, and you can see only some of them on the slide, and I would really like to thank my colleagues that are here and in Moscow, and that is the finish. Thank you very much.
We are open for questions, and all the speakers may respond if that is okay. Questions? No questions?
Thank you so much. That was really interesting. I was just wondering how big you can print these things. So especially like really complex liver bioprinted organoids that are vascularized and all of that, how big can they get?
That's a really good question, because currently it is one of the main challenges to make not only some simple things, but to make them more like real organ. And it is a big problem, because usually if you print something big, the center will be dead in some days because of the absence of the vascularization and the diffusion restrictions, all of that. We have now some ideas how to overcome it through the, for example, this vascularization, the cuvex, the cells, but still they need some time to develop inside.
Yeah.
So usually we print some things with the pores in it, such as the idea is here. So usually we print not the solid layer of the hydrogel, we have some holes in it so that the medium would flow more evenly, and the amount of the restricted regions will be smaller. And in terms of the sizes, I can show you the biggest thing that we have printed, these two, and this is the 60 millimeter Petri dish. So it's like 3 centimeter, I think, 4 around that. Yeah. Also really good question, because this is some minor technical point, but why the holographic printing, for example, is so interesting in perspective. Because if you will print some really dense and big structures, it will take time, and while you print it, the cells will be dead. And for these ones, it was not so big time. I think maybe 4 minutes, around that. So this is quite fast, but still it is not the whole human, I presume.
Your questions? Yes.
So thank you so much. This was really fascinating. I'm Klaus. I'm not from engineering background. I'm from family medicine and primary care, so clinical practice. And I'm just astounded because I'm just also wondering how your relationship is with your lab and institution, with the innovative application possibilities in clinical practice and clinical care. To what degree have you formed a network or partnerships with clinicians in primary care in different disciplines? Because I could just imagine the immensity of the potential applications for this.
Also, another good question. Maybe here I should say some thanks to Peter, because usually he's in charge of making these big connections and communications.
It's not true. It's
Yeah.
not true. Not me. I just connect you together and you work.
Yeah, and then
Give
we,
an example of Stushka. It's very good question.
yes, because clinicians usually are the different set of the people. They have their own goals, the ideas, and all of that, different mindset. And yeah, for example, we have this Professor Svistushkin, who is a clinician itself. So he works with the vocal folds and eardrums, and still he's really dived participant of our biofabrication findings. So we like meet, we discuss some hypotheses, some what should we test, and he helps to make some initial preclinical studies on that. And that is really a good example when I think the person, the clinician, can touch the results of the work and can just take it and place somewhere where his field is. So yeah, but we are trying to scale these interactions to the different clinical departments. And I think we can see in some years maybe, but still for now, yeah, as you can see from the variety of the objects, we're working with the otolaryngologists, like with the ear doctors, sorry, with the dentists, with the orthopedics, also with the skin department. So quite a lot of them. And usually, yeah, if you will talk to each other, you will find some solutions, but you need some person to took you in a room and start talking. Thank you. Yeah.
We have one question from online. Jackie Friedling asked, great, was to say great talk. How long does the tissue remain viable?
That is really can depend, you know. Here we can see the fundamental differences between the cell cultures and some following steps. Because when you culture just 2D cells, it's usually less than a week, and you need to transfer them to 2 petri dish, to all of them. The organoids, for example, are much more dormant structures. We have cultivated them for maybe a month and a half, and they remain viable. They become more compact, dense, and sometimes the necrotic pore inside occurs of, again, because of the oxygen restrictions. If we're placing that in the hydrogel, on the one hand, it is potentially more stable in long term. But on the other hand, the interaction is starting, and usually when we are culturing the bioequivalents for more than 3 weeks, they start to how do I say, interact with the hydrogel, so they degrade it gradually, but still. And with each week, you will have like more cells and more dense thing, but smaller like volume of it. We have not been more than also a month and a half also around that. But I think if in proper conditions, especially in the bioreactor, it can be higher and longer. So also need to work on it as well.
Out of curiosity, what are the approval process before I can use a artificial ear or ear drum?
It's a really long process because we have some different categories of these cell products. And for example, if we use the autologic product, like from your cells, and you can do something with them and put back to you, not leaving the clinic. It would be just okay, just some informed consent of the patient. But if you are going to take some cells out of the lab, it becomes the different category. So it will be the clinical, more complex clinical product. And you need to have the full set of the safety data, efficacy data, like preclinical studies, the some data on pharmacodynamics, pharmacokinetics, the proof for that it does not contain any infection, all of that. So, but on the other hand, it is not like only for you, it is for the whole technology. So once you have all of that pipeline, you just can have some different humans and all working this idea, flow chart, pipeline. Yeah. So.
Any more questions? If not, let's thank the speakers. Everyone for And thanks everyone online.
Вот это.
Вот они
Биопринтинг?
они не под детальные друзья.
Еще не понял. А это?
Это вот то, что хочется.
Надо публиковать.
Да, как раз хочу. Вот это вот здесь. Это на самом деле нужно слайды. Когда из эксперимента не хватает. Здесь куда-то пока.
Результат классный.
Да,
Я да?
работаю.
У тебя язык английский. Ищи.
Bioprinting. Yes, we do bioprinting. Yes, I know. Now I thought of bioprinting. Exactly. So this is different. Almost so said it's testing the non-bioprinting one or the same touch. All the best things. Thank you. Thanks for sharing the lecture with your team. Yes, yes, yeah. I'm so sorry I couldn't. Thanks. The sound was amazing. Yeah, I shouldn't say that. No, I enjoyed it.
It's complex actually because here we use not the then we look into the spirit and it makes it to form for 3 days.